Government NPG College of Science, Raipur. Today, our guest speaker of first session is Dr. Nandini Sharma, and her topic is Species Diversity of Rotifers, Concepts and Calculations. Before we begin with today's discussion, let's have brief introduction of Dr. Sharma's achievements and accomplishments. Dr. Sharma studied zoology at University of Delhi from 1982 to 1995. After a couple of years of postdoctoral research in India and a training course in lake management at University of Ghent, Belgium, she started working at FES Istagala in 1997. Her topics of interest are related to aquatic ecology, mainly cyanobacteria, zooplankton, and fishes. Dr. Sharma has published more than 275 research articles in highly reputed national and international scientific journals, including Nature. Apart from regular teaching duties, she has imparted 40 courses in Mexico and in India also on zooplankton culture, aquaculture, ecotoxicology, and art of scientific writing. Nandini ma'am has worked as thesis director for 65 students at the bachelor's, master's, and doctoral level. She has organized 4 international conferences in Mexico and several national other forums. She has attended more than 90 national and 50 international conferences till date. She has been reviewing articles for more than 50 prestigious journals and member of editorial board of international journals too. In 2012, Dr. Nandini got Scientific Merit Award from FES Istagala, and in 2014, she received Sir Joanna Inez Award from UNAM. This award is being given by National Autonomous University of Mexico to the women who excel in teaching, research, and in culture. At present, she has highest level of national recognition for investigators and highest level of productivity linked bonus, that is Pride D, being offered by UNAM. We are blessed to have such a knowledgeable and learned person as our guest speaker today. So now I welcome ma'am to enlighten us all. Welcome ma'am.
Thank you very, very much for your very kind words. Thank you so much. Let me begin by sharing my screen. Just 1 minute. No, what is it? Why it's not working today? No. Can you hear me? I don't have the option to share the screen. Now it has come. Just 1 minute. 1 minute. I don't know why it's giving me some trouble. Just 1 minute. एक मिनट अब शायद हो जाए 1 मिनट हाँ, अब देखते।
हाँ मैम, अब दिख रही है स्क्रीन आप?
अब ठीक है।
यस।
ये शुरुआत दिख रही है आपको?
क्या मैम?
अच्छा,
आप इसको स्लाइड शो पर डाल देंगे तो स्लाइड शो चालू हो जाएगा।
अब दिख, स्लाइड चेंज हुआ?
नहीं मैम।
स्लाइड नहीं चेंज हो रहा है। स्लाइड नहीं चेंज हो रहा है ना?
स्लाइड शो पर डाल देंगे क्या मैम इसको?
स्लाइड शो पे ही है। स्लाइड शो पे ही है, स्लाइड चेंज नहीं हो रहे हैं।
नहीं मैम, स्लाइड चेंज।
अच्छा, ये मेरे यहाँ पे तो चेंज हो रहा है। अच्छा, मैं फिर से कोशिश करती हूँ, 1 मिनट। अ,
मैम विजिबल था स्लाइड आपने दिखाया था इससे पहले?
ये स्लाइड विजिबल है, पहला वाला, बायोडाइवर्सिटी एंड इंट्रोडक्शन।
अब हट गया मैम, पहले दिखा था, अभी आपने कुछ
ओहो, अच्छा ठीक है, 1 मिनट 1 मिनट। हम्म। ओहो, ये क्या हुआ? शेयर हो रहा है?
विजिबल है।
हाँ जी।
विजिबल है मैम।
अच्छा, अब मैं स्लाइड शो पे कर दूँ ना?
हाँ जी,
अब दिख रहा है?
दिख रहा है मैम।
और स्लाइड चेंज हुआ?
दिख रहा है, दिख रहा
हाँ,
है। यस मैम।
ठीक है, ठीक है, ठीक है, थैंक यू सो मच। थैंक यू सो मच। Sorry for the delay due to technical problems. So today I will be talking to you about biodiversity and methods to quantify. Also, we will look at some of the saprobic indices, and we will look at some of the other indices to estimate the or evaluate the water quality. Okay, so I always like to begin these talks with an introduction to this book, which is an excellent book by Gaston and Spicer. If you do not have it in your library, please let me know and I will see if I can send you a copy of the book. I can send it to Dr. Das and she can share it with everyone. But just let me know if you have this book. This is a very nice book for students in which Gaston and Spicer, Gaston is a great expert on the subject, a diversity of different organisms. He talks about the concept of diversity and different reasons for which this subject is so important and the problems that we are facing with in this regard these days. So to begin with, how did we start with this term biological diversity or biodiversity? It was introduced by Lovejoy in 1980 at the Amazon Biodiversity Center. Then Rosen developed the word biodiversity, but it became very famous by the great entomologist E.O. Wilson. And these people, they introduced the term and then they gave it wide publicity in the different fora, especially in the Rio meetings in 1992. Okay. After that, the Convention of Biological Diversity was set up at the same Rio de Janeiro meeting in 1992. This was signed by 150 nations, and this has become a very stable organization which works extensively on biological diversity in different parts of the world. So there is a very strict definition that is followed by the CBD, that is the Convention on Biological Diversity. It says that biological diversity means the variability among living organisms from all sources, including inter alia, terrestrial, marine, and other aquatic systems, and the ecological complexes of which they are part. This includes diversity within species, between species, and of ecosystems. So this is a very comprehensive definition of biological diversity, which I think students must reflect upon, think about it while doing their research. Does biodiversity actually matter? Of course it does. We depend upon our environment for everything, directly for food and medicine. We also use information for the biological control of organisms. We also use it for generating ideas for the industry. For instance, Velcro was generated by observing the different interlocking system of the seeds of Arctium. Then we have echo sounders from bats. There are numerous examples. For ecotourism, it's very popular. In Mexico, we have this migration of the monarch butterflies, which come over 5 generations from Canada to Mexico every year. And they come sometime in December. They leave in the month of March. Extremely beautiful and extremely interesting phenomenon. So these are the different reasons for which biological diversity, we must study it. The second point is that no organism needs to have a reason to live on the planet. They have all evolved here. They all should be allowed to live. We have no right to make any species extinct. Considering these facts, let us look at other reasons for the biological diversity. For instance, 7,000 kinds of plants have been grown or collected as food. 20 species of plants, just 20 species of these 7,000 kinds of plants that have been grown. Not 7,000 exist, many million exist, but 20 species of these give 90% of the world's food. And only 3 species, wheat, maize, and rice, give 50% of the world's food. So consider these facts, and now it is a good thing that we are trying to, especially in India, introduce other forms of cereal use for human consumption. Now consider this other fact, natural sweeteners. All of us are well aware of the rising epidemic of diabetes throughout the world. You have stevia, which is commonly used now. It was found first in South America. It is 300 times sweeter than sugar. But then there are other plants which are 1,600 times sweeter than sucrose. But we do not know about the use of these plants, about their growth, about their culture techniques yet. Now, when you consider biodiversity, one of the major problems that people talk about is that what will happen when the habitat is reduced to will the biodiversity reduce proportionally? Now, when the habitat of an animal is reduced to 1/10, what will happen? Actually, just by 10% loss of habitat, there will be a 50% loss in the number of species. This was proven by Wilson in 1985. And this is the fundamental problem which many people, mostly politicians, do not understand. Just by reducing 10% of protected area, you will have a tremendous loss in the biodiversity. So these points must be taken into consideration when we are studying the subject. Now let's look a bit at this concept of megadiversity. Megadiverse is a specific term. It is not that all countries are megadiverse. You can see on this map, a few countries are megadiverse, and there are 2 criteria for a country to be declared as megadiverse. Number 1, it should have at least 5,000 species of endemic plants. What does endemic mean? Endemic means that these plants do not grow in any other part of the world except for the zone in which they are found. And these megadiverse country should also be bordered by marine ecosystems. So inland countries, for instance, Mongolia or Czech Republic, they are not considered as megadiverse because they do not fulfill these two criteria. Am I going too fast? It's okay?
Yes, ma'am.
Thank you. Okay. Now, the second term, which many of you may be familiar with, is the biodiversity hotspots. You can see over here much of Mexico, and this is a very sad situation actually. It recognizes areas which have 1,500 species of vascular plants not found anywhere else on the earth, endemic species. And the area has lost at least 70% of its primary native vegetation. So it's a very sad thing to be recognized as a biodiversity hotspot. But we do have biodiversity hotspots. Much of Mexico comes under this category. And if you notice over here, this western coast of India, especially the Western Ghats and all this region, also is considered as an important biodiversity hotspot. Very interesting articles have been published recently, I think last month or this month in Nature on biodiversity. And it would be interesting for you to read it. If you do not have access to it, please let me know and I can try to send you this article and the book. Okay, so now we come to these terms. Alpha diversity, what does it mean? Suppose let's restrict ourselves to zooplankton and rotifers. For instance, if you study the rotifers in a particular water body, you are studying the alpha diversity in that habitat. And if you study different groups of organisms in that area, for instance, trees and butterflies and insects and whatever, birds, then you are trying to estimate the gamma diversity, which is the total species diversity of a landscape. And beta diversity is considered as the proportion between this total diversity and diversity of a single group of organisms. So these are the three concepts of diversity, and these are the three measures. Most studies base their research and base their analysis on alpha diversity. I wonder if you have read this book. We all know the importance, the loss of species, the extremely rapid loss of species. And Elizabeth Colbert got the award for this, the Pulitzer Prize, when she started talking about the sixth extinction. What are the threats? Why does this happen? Number one, and this has been named as the evil quartet by Jared Diamond in a study way back in 1984. He said that what are the threats to biodiversity? Number one is direct exploitation, habitat loss and degradation, the presence of introduced species, and extinction cascades. So if you affect one species, you will affect many others who depend upon these for their survival. So these are different reasons for which, biodiversity is lost. All these examples are given on this slide. And perhaps I'll just take a moment to explain this one, which is about the, oh, oh, I don't know how to go back. Uh, yeah. So in this slide, you can see a condition that was found in New Zealand. So there you have the Haast eagle and the moa bird. Now, the Haast eagle depends upon the moa bird for its food. When the Maoris came, the tribal people, when they reached New Zealand, they started hunting the moas because the moas had no defense. They never evolved any kind of defense because they never faced any kind of predation. So the moas were hunted down by the Maoris, and the Maoris almost made them extinct. And as soon as the moas declined, the Haas seagull also declined. So this is what is known as an extinction cascade. So if you just affect one species, you will affect a whole lot of species associated with that. This is another concept which is known as the sentinel extinctions, that species become extinct even before they are studied, even before they are described. This was described for the first time by Gentry and Dodson in 1978. In Ecuador, at least an area with 90 endemic plants. That means those plants are not found anywhere else. Imagine, just try to reflect for a moment. Imagine the loss for mankind or for the planet when species that have evolved and are not found in any other part of the world, they were all cleared for creating agricultural land. And this is the fundamental problem. This is why we need young people to be aware of these concepts and to work on them in small measure. Each person does a little bit with any group of your interest, but it is very important to continue with this kind of research. Because if not, we will lose millions of years of evolutionary effort. Now, coming to the point about how should we study the diversity of rotifers in one single water body or of zooplankton. My suggestion to all of you is collect samples. So you know how to filter water, you know how to fix the sample in the field, and you should do like it was suggested yesterday. You should collect from 3 or 4 or 5 different sites so that you have some replicate understanding of the water body, and you should study it for all the seasons of the year. According to Lampert and what's his name? Hakumat Rai. Lampert and Rai, they suggested that every 15 days water samples should be collected. But if it is difficult because of logistic reasons, then collect it every month from 3 to different sites, as I suggested before. Identify the zooplankton before you start counting them. And then you should start quantifying the zooplankton with a Sedgwick rafter cell. A Sedgwick rafter cell allows you to estimate the density exactly in 1 milliliter of your sample. And then you make the calculations about the density of the zooplankton. If you have any doubts, please let me know at the end of the talk. Now you determine the diversity and water quality using different indices. If you do not have a Sedgwick rafter cell, you can do what we do with our students in the BSc classes. 1 milliliter of water is distributed into various droplets on different slides, and count how many individuals of each species you have in each droplet. You add it up and it will give you the idea about the density of the zooplankton per milliliter of your entire sample. You have to obviously change this to individuals per liter. If you have a doubt, please ask me at the end of the talk. Okay. Now you calculate the Shannon diversity index using this formula. It is very important not to forget to put this minus sign at the beginning of the index, because when you have a log natural of a decimal, it will definitely be negative. And then that negative and this one will cancel out and you will get a positive value. Now, you can use log natural, you can use log to the base 10, or you can use log to the base 2. These are the three that are commonly used in the literature. So if you are comparing your data with somebody else, pay attention. What is the exact formula that they used to do the analysis? Because you will have to do it with that exact formula if you think that the manuscript is very important for your research. There is another index, which is the Shannon Equitability Index. And what happens with this one is that you have to use this formula, which is the species diversity index divided by the log natural of the total number of species. So it tells you that it ranges from 0 to 1. And 1 indicates complete evenness. It tells you if there is a dominance of one group or if all the groups are evenly distributed in that system. You also have a Simpson's diversity index. This is the one that is normally used. Actually, this is an index of similarity. So the 1 minus D is the index of diversity. These are the calculations. So for your convenience, I'm explaining the species diversity index over here. So you have different species. We just put a small example, and these are the densities. Now you calculate the total, and 500 divided by 610 gives you the proportion. 50 divided by 610 is the proportion of the species in the sample. Now you find out the log natural of this proportion, multiply your proportion by the log natural of the proportion. It's a simple formula. And you get the sum of all this gives you the diversity. The same thing is followed over here, but notice over here that the density of the organisms are much lower, but the distribution is more even. So what happens? Your diversity index tells you that the diversity is low here. It is not low because of the density of the organisms. It is low because of the dominance of one species. And here the diversity index is high. This is a wonderful page. You can take a photograph of this slide, and it is very, very useful for quickly calculating biodiversity. And it works extremely well, very easy to use. So please take a photograph and try to use this page for calculating biodiversity. Can I change the slide now?
Yes, ma'am.
Thank you. Now, here is the index that we were talking about yesterday, about the saprobic index. You have it over here. This is the formula. You need to find out the relative frequency. 1 indicates that the species is uncommon, 3 that it is common, and 5 that it is abundant. So here you can see a small example. What does it say? You have a density of 500 of Brachyonus caliciflorus. Trichotria tetractis is another rotifer. You have only 12, 18. Brachyonus angularis 100 and Asplankna sieboldii 8. So you have 5 different species and their densities. Now, based upon this description of the frequency, we put 5 for Brachyonus caliciflorus, 3 for common in Brachyonus angularis, and rare we put Trichotria tetractis, similis, and Trichocerca similis and Asplankna sieboldii. Now you found this valence in the article by Sladicek in 1983. It's another very good article to do these calculations. If you don't have a copy, let me know, and I will send you a copy of this through Dr. Das. So you have this valence values, you multiply them, and then you follow the formula where you have to just divide this total divided by the total of the frequency. And what did we find? We found that according to this, density of rotifers and this diversity of species richness of rotifers, the water body is beta mesosaprobic, 1.9. So this is exactly what I was telling you yesterday, that these values, this type of research is extremely good. It has got a lot of application, especially when you are talking to agencies who want to know about the water quality for different uses, like was mentioned yesterday, for bathing, for drinking, for agriculture, for whatever. So these indices are very, very useful in this regard. Then there is this other index which we were talking about the trophic state index proposed by Esmond Caraban. The formula is very simple. You have it over here. It's simply 5.38 log natural of the total rotifer density. You don't even have to know what species they are. You just have to count. You just have to know if it is a rotifer or not. Count the number of rotifers, multiply it 5.38 by log natural of n, and add 19.28. That's it. And you will get the value. Now, if you see over here, this small example, when there were 10 individuals, the water body is mesotrophic. If there are 120 individuals per liter, it is mesoeutrophic. And if there are more than 1,000 individuals of rotifers per liter, it is a hypertrophic water body. So these are the things that you can easily do, very simple tools for you to estimate and for you to describe the water quality of for different agencies or for your own research, for your own interest. Thank you very much for your attention. If you have any questions, I'll be happy to answer them, or else we can move on to the next talk. But I think that we should discuss this talk first.
Ma'am, my one question is here.
Yes.
How introduced species disturb biodiversity?
I didn't understand your question.
Ma'am, there are so many reasons for the disturbance of biodiversity.
हाँ, तो इंट्रोडक्शन कैसा होता है?
इंट्रोडक्शन से कैसे डिस्टरबेंस होता है बायोडाइवर्सिटी से, इंट्रोड्यूस जो स्पीशीज हैं,
हाँ,
हाउ इंट्रोड्यूस स्पीशीज डिस्टर्ब बायोडाइवर्सिटी.
अच्छा, ठीक है. अब मैं समझ गई. 1 मिनट में I'll try to go back a little bit because I would like to talk to you about this example to answer the question. Can you see this fish here? The scorpion fish?
Yes, ma'am. Yes, ma'am.
Okay. Now, the scorpion fish is a fish that belongs to the side of the Indian Ocean from Malaysia, from that area. Okay. Now,
Yes, ma'am.
now this fish has reached Mexico. It is now an invasive species in the Gulf of Mexico in the Caribbean.
Yes, ma'am.
Now, what happens? This is what happens with invasive species.
Yes, ma'am.
In its native habitat, the fish has evolved and predation and the prey, the interaction has co-evolved. So the fish
Yes.
has got predators. So
Yes,
the population is kept low.
yes.
In
Understand.
Mexico, the fish is new to the habitat. It
Yes.
is a voracious predatory fish, but there
Yes.
is no other organism that has learned how to feed on this fish.
Okay.
Because the fish has got two very strong defenses. Number one, you can see all these spines. And
Yes.
number two, it is toxic. So there is no organism that has evolved to consume this
Control.
fish in the new habitat.
Yes.
Now it will bring down the density of all the native species that live over there. So this is what happens with invasive species. The same thing happened with Methotriphes in Canada. It is a European species. In Europe, it has got predators. It was introduced into Canadian waters. There are no predators. It brought down the density of zooplankton drastically, and therefore fish production declined because there was no food.
Food, yeah.
So these are the reasons how the invasive species affect the different habitats. That's why you should be very careful. That is another very important reason for monitoring the diversity, so that you know immediately that this species does not belong to this part of the world. It belongs to another part and it should be carefully monitored. For this, it's a very nice question because it lends it to for discussion and for reminding me of some points. There is an excellent monograph by Hendrik Segers. He's an expert, global expert on rotifer taxonomy. And this excellent monograph is there in the journal called Zootaxa. So it's another copy. If you do not have it, let me know and I'll try to look for it. So Zootaxa, it talks about all the rotifer species and
Yes.
two very important things. Number one, the correct name, the correct author's name of each species. All the synonyms have been excluded.
Yes.
And the second point is it also talks about the distribution.
Yes.
So when you are with Dr. Sarma, please ask him, because there are species which are equivalent in rotifers to Asia and America, for instance. So these species cannot be found, and if they are found, they are either introduced or invasive. You should be very
Yes.
careful. So Dr. Sarma, I think, will be talking about this subject a little more with all of you.
Yes, ma'am. Thank you very much, ma'am.
Thank you.
Thank you.
Any other question?
No, ma'am. Thank you.
Okay. Any question from the students? I really appreciate it if the young students ask.
Good morning, ma'am.
I have a
Hello.
if you see,
Hello.
Hello, yes. I
Yes.
can.
Yes, ma'am. Good morning, ma'am. I'm
Good.
Anuradha Sarma, research scholar.
Uh-huh.
Yes, I have one question, ma'am.
Yes, please.
Have we have any techniques which will be used to restore biodiversity?
It's a very tough question, very difficult. The point is that you need to first improve the habitat. So the point, that's why, it's very important first to study all the habitats carefully. It's very important to keep an eye on the quality of the habitat, quality of water, for instance. And the second point is on invasive species, because there is a very small window of time in which you can work to remove a species. After that, it establishes itself in the system and it's very difficult to eradicate. So these are the points that you should consider for answering that question. But it is a very difficult one. Yet there are very, not numerous, but excellent case studies where they have had a lot of success in improving the habitat, in improving the diversity.
Okay, ma'am. Okay, thank you, ma'am.
Thank you.
Madam, I have a simple query.
Yes,
You see your last slide, you have given a data.
yes,
You have identified the mesotrophic, mesoeutrophic, and hypereutrophic on
yes,
the basis of number of species.
yes.
Do we have similar kind of number of species to identify with the other groups? And this is for rotifers you have suggested.
Yes.
For other groups of zooplanktons or for other groups of phytoplanktons, do
Uh,
we have similar kind of number to
they
classify bodies into these groups?
Yes, there must be. There must be some kind of, this type of data which is available. If phytoplankton is your interest, there is a very nice book on freshwater algae by Bellinger and Siggy. If you have the book, you can use it. If you don't have it, let me know. But in that book also, they talk a lot about using algae for determining the water quality. This TSI rot is on rotifers. The TSI, the trophic state index, is also based on chlorophyll A, exists.
So the rotifer number which you have suggested,
Mhm.
uh,
Can you please speak louder? I can't hear you.
Madam, the data which you have shown
Uh-huh.
about rotifer,
Uh-huh.
classification of water bodies. It is the reference is available with you.
I can't hear you. I can't hear you. I lose you. Please stand closer to the mic. Yes.
My question is that, madam, in last slide,
Mhm.
regarding rotifers numbers,
Yes.
you have identified the type
The
of
density. Yes.
it, how this number has been taken, and which body or which reference.
No, this is just an example. This is just an example to show you how to calculate it. But when you go to your own water body, you will calculate how many rotifers are there, and based upon that, you will find out the quality of water. This is just an example. This is not a value, actual value.
So I am looking for a similar, if any such numbers are available to
Yes.
identify water bodies, then it will be easy for the researchers to just go through the identification and the number in
Yes,
to
there are
identify particular water body.
correct. There are many articles. There are many articles on the subject, and we just need to know. If you need any such help, please write to us, and we will send you some papers.
Okay, okay, thank you.
Thank you. Thank you. So should I continue with the next talk then? Because I think Dr. Sarma also needs some time. I'll try to
Good morning,
do.
ma'am.
Good morning.
Ma'am, I want to ask, what are the challenges occur for studying biodiversity of species of rotifers, and what are the environmental factors that affect rotifers species biodiversity?
Okay, what are the challenges? Number one, you need to know rotifer identification and taxonomy very well. Your data must be completely reliable. The best way to do it is, Dr. Sarma will be talking to you more about it, but confirm each species with a good expert. And these days, people are willing to help young students. Send your photographs to an expert, confirm the species, and then continue to count it. After that, you need to have, this is the number one problem. The second is to plan your work very well with the clear hypothesis, clear objectives in mind, and then start collecting the data. And what was the second part of your question?
Ma'am, what are the environmental factors?
Environmental factors. All the environmental factors, for instance, temperature, salinity, conductivity, nutrients, all of them will affect rotifers eventually. So you will find a difference, pH, you will find a difference during the whole year, and the rotifer diversity and density will change. There are biotic and abiotic factors. So these are the abiotic factors, and the biotic factors are competition, predation, facilitation, parasitism. All these factors will also affect. So you have to be very careful about how, first you need to study these concepts well. So there is a very nice book called Limno-Ecology by Lampert, by Winfried Lampert. So you study that book well, where he gives all the examples of these interactions based on aquatic organisms. And then you will find that all of them affect your rotifer. So the rotifer density and diversity changes dramatically during a year.
Yes, ma'am. Thank you, ma'am.
Welcome. So
Can you please your PPT, sir, so
yes,
that can be very helpful for our student research scholars also?
I beg your pardon, I didn't get the first part. There was some disturbance.
Can we have your PPTs as a study material for our research scholars?
Of course, we will send it to you.
Yes,
We
sir.
will send it to you, and we will also send you the books and articles.
Thank you so much, ma'am.
You're most welcome. You're most welcome.
Then you can continue with your next lecture.
Okay, thank you so much. I'll start that now. So let me just give me a moment, because this thing always gives me trouble here.
ma'am.
Can you see the screen?
Yes, ma'am. It's visible.
And
Yes, ma'am.
is the slide, did the slide change?
Yes, ma'am. Yes, ma'am. It's changing also.
Yes,
Okay.
ma'am.
Okay, excellent. Okay, perfect. So now I will talk to you very quickly about plankton culture techniques, so as to leave more time for Dr. Sarma, because that session is very, very interesting and very important to learn how to identify tips to identify rotifers. So I'll talk to you very quickly about how you can culture plankton. One young student asked about this yesterday, and it's an interesting idea if you can try to establish it. So let's look at the importance of culturing plankton. First of all, plankton plays a very central role in the food web. It has a wide distribution and it is used to verify different ecological hypotheses for different applications such as ecotoxicology, aquaculture, and for taxonomy and systematics. So all these reasons make it important to culture zooplankton. Well, to begin with a zooplankton culture, you must have the food. So the food that we normally use is algae. Algae we normally use in freshwater systems, Chlorella, Senadesmus, Chlamydomonas, Cryptomonas. For marine systems, you can use Tetraselmis, Nanochloropsis. Many different types of algae are used, but try not to have very large ones. You should have algae which is 5 to 10 micrometers in size. It's ideal for the culture of rotifers. What culture medium we can use? We can use different kinds of media such as Poole and Lorenzen or Bowles basal medium. It's not important because most of the media are practically very similar. They have micro and macronutrients. And in one study, they showed that 200 species of algae were cultured in just 2 different media, one for freshwater and one for marine. So use any one medium. You don't have to use all of them. You can see the formula. This is given in the literature. You can collect all this from the literature. There's no difficulty in finding this. Different types of medium. What are the different salts that we need? So we need all these macronutrients and we need several micronutrients. Prepare this very, very carefully because this is the basis for excellence in algal culture. And the basis for excellence in zooplankton culture is the quality of your algae. So this is what I was telling you. Mostly they have macro and micronutrients. The macronutrients are like potassium, sodium, calcium, etc. Micronutrients, boron, zinc, cobalt, copper, cobalt, very small quantities. But the point that is very important is to add periodically carbon. And we normally use sodium bicarbonate as the source of carbon for our algae. Now, there are 3 different phases in algal growth: the log phase, the exponential phase, and an asymptotic phase. So let me go to the next slide. Yeah. So you have here, it is just picking up. Here is the log phase, and then it reaches an asymptote. When should you harvest your algae? When it reaches about this phase over here. How can you quantify the algae? Using a Neubauer chamber, which is used to quantify blood cells. So that is the same slide which you use to quantify algae in our lab. And then you harvest the algae. Once you harvest the algae, you can come here. And how do you sediment the algae? There are 2 ways. If you use the entire medium, your zooplankton will not grow well. So you must sediment or centrifuge the algae. So for sedimentation, we normally put it into the refrigerator. And for centrifugation, if you have a nice centrifuge, you can centrifuge the algae and get the concentrate. Then if you have large outdoor cultures also, you can do a similar technique, but do not allow your algae to reach beyond this phase for harvesting, because after that it starts to develop toxins. So even if there are more nutrients in the medium, don't worry, but use freshly collected algae for excellence in rotificulture. Now, how can you do this over here? You have all this algae in a small tube. You scale it up with the medium. When it reaches a high density and a volume like this is about 15 milliliters, then you go to 50 ml, then you go to 250, you go to 500, 1 liter, 5 liters. Now you have sufficient algae to inoculate in outdoor tanks. Now, if you try to inoculate from this small test tube to an outdoor tank, there are too few cells for the algae to compete well with other organisms. So you will fail. You should always have a big inoculum to start up your culture in a large container. So this is exactly what I was telling you. Here, the point is that you should try to use a little bit of aeration. You can use the small filters for aquaria, which are used to give air for the fish. You can put the same thing for the algae. The reason is not because algae need oxygen, but because all the cells will be in circulation and they will get sufficient light. You must have a constant source of light. And if you have a lower temperature, 15 to 20 degrees, algae grows better. So think about it, a air-conditioned room and a constant source of light, aeration, a proper medium, and a proper inoculum, and you will have success in algal culture. What are the different kinds of cultures? There are non-continuous, there are continuous cultures. We normally follow the batch culture system. So we inoculate the algae, like you can see in these tanks in our lab, and then they reach higher densities over here in large volumes. This is 20 liters, this is about 1 liter. And once you reach a very dark here culture, then you start sedimenting it, use it, and throw away the supernatant and start the culture all over again. There are different problems in algal cultures. You need to have proper light, proper aeration, proper medium. You can also have sometimes some contamination with bacteria. Then you can use perhaps some antibiotics. Read the literature, you will find the information over there. And if you have a growth of unwanted zooplankton, such as deltoid rotifers, you can use different mesh sizes to separate the algae from the zooplankton. For instance, if you have a mesh size of 20 micrometers, you can easily separate chlorella from the deltoid rotifers. There are also certain media which have been described. Some people use it. That is a combo medium, which is the same freshwater medium for the culture of algae and zooplankton. This also may be useful for you. Please try. These are the meshes I was just talking to you about. So you have these meshes. If you do not have these large conical ones, you can always have these small ones, which are made in our lab by cutting two pieces of PVC tubing, which is available here or everywhere, and put a mesh in between, stick it up with araldite or with some kind of glue, and you will have a mesh ready to use. You can have these size of meshes, or you can have small meshes, depending upon your needs. Now, zooplankton cultures, these are extremely important, particularly for aquaculture. So if you are capable of culturing zooplankton, you will definitely have a value in the job market, because you will be able to produce food for fish larvae. And that is the fundamental point at which you have a great loss in fish larval production. So to start a zooplankton culture, you need a good microscope, you need pasture pipettes, a few meshes, plastic containers or glass containers, and appropriate food. That is the fundamental, only 5 things, very important. Culture media and appropriate food. We normally use the EPA medium. I'll give you the formula in a bit. And this is how you start the cultures. You identify the zooplankton, you separate them with a fine pipette, take them from a local water body, clean them. Cleaning means you pass 20 or 30 individuals, 50 individuals repeatedly through clean water. So you put 50 first from the medium, from the lake, into a clean vessel. Then count these clean individuals, put it again. So now you will be separating and you will have very clean rotifers or any other species of zooplankton to begin your culture. Always try to look for females with eggs. The culture will grow fast and you will be very happy. You need good quality water. So this is the medium that we normally use, the EPA medium. This is the formula and you prepare it with distilled water and you can keep it for a week, 10 days, and you can use it for your cultures. So we use distilled water with these 4 salts, very easy. And we maintain, I have written here 40, but I think we have cultured more than 80 species of zooplankton by now. So rotifers and cladocerans, like Dr. Sarma showed you yesterday, they reproduce mostly by cyclical parthenogenesis. So this parthenogenetic reproduction allows them to reach extremely high growth rates. Brachyunus caliciflorus is considered as one of the fastest growing metazoans on the planet with extremely high growth rates. Moino also has very high growth rates. It's a cladoceran. And these 2 species are very important for aquaculture. Copepods are interesting. They are also important in aquaculture, especially in marine systems. However, the growth rate of copepods is slow because copepods have different stages of development. So they have 6 naupliar stages, 5 copepodite stages, and each one takes at least 1 to 2 days. So this slows their growth rate, lowers it. Copepods are very good food, however, they are for 2 reasons. Number one, in freshwater, in marine systems, cladocerans are very few. So copepods are very, very important. Fish are genetically, they have the program to eat these organisms and their food quality is extremely high. They have very high levels of omega-3 fatty acids. Never mind whatever diet you give them, they are capable of synthesizing this in their body. And that is why copepods are very important also for aquaculture. If you have copepod cultures, they tend, some species, especially in freshwater systems, they tend to be cannibalistic. So you need to use a container like this, and this type of container with a mesh to separate the adults from the nauplii. So you will have the adults over here, and the nauplii will be able to escape. And when the nauplii escape, you will have a higher growth rate. Otherwise, the adults tend, some of the species at least, they tend to eat their own nauplii. Cladocerans, how do you culture them? As rotifers, you can also feed them algae and different types of mixtures of algae. Try not to look for blue-green algae or cyanobacteria as they are known now. These are not very good quality food for zooplankton. If you have males in the system, like you can see over here, this is a rotifer female and a rotifer male. It looks more like a ciliate. This is a cladoceran female Daphnia, and this is the cladoceran male Daphnia. So there are some differences. I think Dr. Sarma spoke to you about it yesterday. I won't repeat it. But the point is that if you have males in the system, eventually you will find resting eggs and ephipia. Do not throw them. Filter it and keep it so that you can have a stock, because the ephipia, even if it is dry, keep it in the refrigerator. After 3 months or 6 months or whenever, try to put it in water with a heat shock, a light shock. That means that if it is in the fridge, put it quickly at 20-25 degrees, put it in the sunshine, put it in front of light, and some individuals will definitely hatch. And then you can start your culture all over again. So you don't depend upon anybody. This is what I was telling you about the resting eggs or the ephipia. This is the structure that we are looking for. When you see the cladocerans with this, the growth will be going down, but keep these ephipia. They are very useful. What type of diet we can use for culturing rotifer? You can use yeast, bacteria, algae, organic waste. We also use a lot of nehayote, which is the waste from the tortilla industry. But here you can use different kinds of organic matter, and rotifers should grow. But algae, of course, is most standard and gives you the best results. Now, if you want to feed your rotifers in fish ponds, let me see. So you need to add different types of fertilizers. We add liquid fertilizers, powder fertilizers, or farm or poultry waste. You can use it. You can leave, for instance, a little bit of cow dung, a little bit of poultry waste in water. And within 3 to 4 days, you will have a dense algal growth. Filter that water with the algae, and you can use it as food for your rotifers. But be careful for contamination due to parasites or other problems. Now, why do we need so many rotifers? You can see in this slide that one single fish larvae needs at least 100,000 rotifers during the first 2 weeks of its life. So this is the point. When the fish larvae hatch, you need to have extremely high density of rotifers. The species which are most commonly used for aquaculture are Brachyunus plicatilis, Brachyunus rotundiformis, Brachyunus demediatus, Brachyunus caliciflorus. These are the commonly used taxa for marine and freshwater systems, and try to culture them in high densities, but please plan your cultures, because there is no point in culturing 1,000 liters of rotifers if you don't have any fish larvae. They will grow and then they will die. So depending upon when you need these rotifers, invest in the culture. The nutritional value is very high. You can see over here, Artemia is very expensive, but Moina, which is practically free, has better nutritional quality because it has no ash. Similar is the case with copepod. So this, if you develop this culture technique, if you learn it, it is extremely useful. Consider the nutritional value of rotifers with different diets, with yeast and with chlorella. You can see that the protein content of the rotifers goes down on a diet of yeast. So yeast can be used to maintain the species, but eventually you should feed them algae before giving them to the fish. What are the problems that we can face in cultures? You can have different types of contamination due to ciliates, other types of protozoans. You can have problems due to predatory rotifers such as Asplankna, or due to other predatory organisms such as flatworms. But if you keep your cultures clean, if you change them with in a regular basis, and if you are careful and keep observing the cultures, you will not have these problems. And if you do, you should be able to separate these predatory organisms or the others through meshes. Otherwise, you have to begin the culture all over again. So this is the Asplankna, which is a predatory rotifer. It can be quite problematic. So be careful, because if it enters a culture of Brachyunus calciflorus, within 2 days, you will only have Asplankna. Asplankna is jelly-like rotifer. It does not very good food for the fish. So this is not very useful for you. Thank you very, very much for your attention. And I'll be happy to answer any questions that you may have. I know that this talk was very fast, but it was just to give you an idea about how rotifers can be cultured. And you need to practice. And while practicing, if you have any problems, please do not hesitate to write to us, and we will be happy to answer you. Any questions? Or maybe you can start the session with Dr. Sarma and ask any questions there also.
Good afternoon, everyone. There is a question.
Yes.
Madam, how long this culture can be made viable in laboratory condition? One thing. Secondly, all these zooplanktons, they are herbivorous, or they may feed upon other smaller zooplankton during in culture.
All of them are not herbivorous. There are some which are carnivorous, and then you have to offer their prey also. So you have to culture the species of interest plus the prey. And for the second part of your question, how long you can maintain it? It depends how carefully you maintain the organisms. For instance, we have one species of copepod which has been in our lab for 19 years now. And we have some rotifer species that have been there for several months. So it's not difficult to maintain them. For instance, we have 4 years, 5 years. It depends upon how you maintain these organisms.
Suppose
It's not. Mhm,
we want to maintain it in laboratory condition,
mhm.
would we have to supply the algal food regularly or any other thing to be required to be
No,
maintained?
you have to change the culture. You have to change it using a small mesh, put fresh water and fresh algae. Because if you do not change the culture, it's called changing a culture. That means that you take the culture, you filter it, you pass to a fresh medium of algae and water. Because if you keep it in the same medium, it will develop, it will accumulate all the toxins. So that has to be done in the case of rotifers at least once every alternate day, at least, if not every day.
Is it possible to make their monoculture?
Yes, yes. We have monocultures. You start with one single individual and it's known as a clone as long as males do not appear. Otherwise, it's a monoculture. Yes, it's very easy to do.
Okay, thank you, madam.
Thank you very much for your questions.
Any other question, please? Good morning, ma'am.
Good morning.
Ma'am, can we use the same rotifers for all the carp species of fish?
Yes, yes. You normally you can use Brachynos caliciflorus for freshwater species and Brachynos plicatilus for marine species of organisms. And you can use the same species for all the fish. There's no problem about that.
Or it is changed according to
No, don't change the species, because if you are interested in fish, don't change the rotifer species, because then you will have contamination with other rotifer species, and then you will have problems of competition and exclusion, and all these things will also come up. So use one rotifer species.
Thank you, ma'am.
Thank you.
So, a heartfelt thanks to Dr. Nandini Sharma ma'am for her excellent lecture on species diversity of rotifers as well as plankton culture techniques and collection of samples, concepts and various calculations. And of course, the interaction session proved the interest created among the audience. And thank you so much, ma'am, on behalf of everyone present here for this wonderful presentation. Thank you so much.
Thank you very much. Thank you.
There is a break of 15 minutes. So exactly after 15 minutes, please be here so that we
Yeah,
can continue.
because that is a very important and a very, very interesting session with Dr. Sarma. He will give you very valuable tips. Do not miss it. Come back.
Yes, ma'am. Thank you.
Thank you so much. Thank you.
Chalo, be here within 15 minutes. Okay.
Thank
12:30,
you.
everybody should be there. Okay, thank you, ma'am.
Thank you.
Good afternoon to everyone. We start after the break. And I am going to introduce Professor Sharma sir. Sharma sir is an aquatic biologist known for his research contribution in the field of freshwater nutrition. He is full-time professor of the National Autonomous University of Mexico since 1995. He is elected member of Mexican Academy of Science, International Society of Immunology, Royal Society of Biology, London, and National Researcher of He has published more. He has, pub- he has published more than 300 articles and book chapters of his 285 scientific works are from Asia Index Journal, including Nature. And he is currently editor of Rotifers News Virtual Newsletter. Today he will discuss about the topic of identification of rotifers from different sites. Then I welcome to Sharma sir to deliver this topic. Thank you so much.
Thank you very much, Madam, for your kind words. I will start a very brief description of what features should be considered uh, for rotifer identification. For this, I will share the screen. Can you see the screen?
Yes, sir.
Can you hear me as well, right? Okay. So first thing is very important to identify the species. Before you do any work, it could be ecology, it could be ecotoxicology, aquaculture, for any purpose, any species that you are culturing, you should know what species you are culturing. So there are experts. So when you hand over the species, they will identify for you. However, that is not sufficient because you need to know also what species you are culturing because it is your own species under consideration. So what you have to do is first thing is
Sir, please share the slide mode, slide show.
it's not in the slideshow. It is in my okay. I'll put again. Um, it is still not in the slideshow. Um, yeah. Can you see the full screen? Not. I will stop and again. Okay. Share the screen. No. Is it okay now?
Not yet, please.
But my screen shows it is in full screen. But okay. Can you see it? Okay.
Continue.
Okay, so the first step is rotifer collection. So how are you going to collect the rotifers? You need to collect the rotifers using 40 or 50 micrometer mesh. You can also use a little bit larger, but smaller species like Anurops fissa you will not be able to collect, especially for quantitative analysis. There are species which are as small as 40 micrometer or even smaller in width-wise. So it's better to use 40 to 50 micrometer mesh is sufficient for you will collect about 95% of the rotifer species in a given water body. As I explained yesterday, you can collect repeatedly in different parts of the water body and also in during different seasons to have very high diversity. Once you have the collections, now observe the sample first under the microscope, live samples under the microscope. Why? It is important to characterize, to detect certain features. For example, the corona structure, the foot length, the toe length, the movement. All these factors are important to determine the species. Therefore, you need to have live cultures, live specimens to observe for some time before you fix them. Well, in most cases, we do not bring the samples to the laboratory, live samples, because it is difficult to transport. So you fix them there and then bring them to the laboratory. Or in case you are interested in, if you are interested in some background noises are coming, I don't know from where. In case you are interested to study molecular biology of the species in question, well, you can fix them in the laboratory. You can fix them in the field with alcohol, 99% alcohol. The general idea is that you have to use the cold alcohol to detect maximum, to collect maximum DNA content. Now, once you have sample, fix it in the alcohol, but do not forget to fix sample also in formalin, because in order to identify the rotifer species, formalin fixation is very important because formalin will give the exact structure of the lorica or the body of the rotifers and the spine lengths and so on, because these are important for determination of, for detecting the identifying, confirming the species status of different taxa. Now, for certain species, not definitely for Brachionus and Keratella, for other genera, you need to separate the trophi, especially the members of the Notomatid as a family. All of them need trophi separation. trophy is about 25 micrometer in size on an average, and is difficult to separate the trophy without adding either sodium hypochlorite solution. This is a commercial grade. You can also get 40% solution, but this is carcinogenic. Generally, industry does not supply unless the demand is specific for a particular purpose. But otherwise, 78% this is normally you use in the house for cleaning the floors and all. This sufficient to add. What you need to do is collect only one individual in one slide with a small quantity of water, but keep it under the microscope continuously. Usually between 10 times 10 into 10 is obviously or even 20 to 10 if you have this kind of magnification. Compound microscopes are necessary for this. Stereo microscopes do not have that much resolution, so it is necessary to keep the specimen under the microscope, live or fixed material. Add 1 drop of sodium hypochlorite solution. It will dissolve all the muscles, and what is left over is simply the structure of simply different parts of the trophy. These are hard parts. They have also a small segment of chitin, and they do not get dissolved easily with this concentration of sodium hypochlorite. Once you have this trophy under the microscope, you can observe them carefully and draw the diagrams. For drawing the diagrams, it is important if you have microscope fitted with a camera lucida. If not, no problem. Take regular photos with normal cameras. You can also use your cell phones. Cell phone camera also is really useful for taking the photos. Okay, so this trophy separation is important for several genera of rotifers. Now, if you want to study the corona structure, how it is, some of the slides I showed you yesterday, that the corona is very widely expanded. To get the expanded position of the corona, Edmondson suggested to use hot water. So you collect zooplankton in a small petri dish, and then boiling water pour suddenly on the top. So what will happen? Those that receive the hot water, the muscles immediately expand and the cilia get expanded. Then this will be useful for identification of some species, especially if you are interested in the corona structure. Now, electron microscope is not needed in most cases, but if you have, it will have a better resolution, obviously. It is as a rule, for zooplankton guides, they are not based on the characters that are not visible by light microscope. It means for identification of the keys, zooplankton or even rotifers are identified using the keys. The standard keys are produced by observing the characteristics that can be visible only under the light microscope. If a feature is not visible under light microscope, this is not useful for identification of the for the construction of the species keys. So this is also important. But what we need to study in order to identify the species, first of all, external morphology. How small or how big is the animal? And whether it is rounded, spherical, it has got spines. If spines are there, how many spines are there, both anterior and posterior, and dorsal and ventral plate structures. Dorsal plate, if it has got any ornamentation, it is very important for Lecane, the genus Lecane, and other species also. Dorsal and ventral plates are separated by a membrane. In order to detect this structure, you need to rotate the animal in different orientations that to get the cross section. For example, Lepidella, Euclanis, all these are identified based on the width of the animal. That is how the dorsal plate and ventral plate are separated by a membrane. How thick is the animal? So for that cross section is needed. It using a dissection needle under the light microscope. You can move the animal laterally, or you can move the dorso ventrally. Then you will detect the the structure of the dorsal plate and the structure of the ventral plate. Now, trophy morphology, in some cases, the trophy is asymmetrical. So some parts will fall out very easily. Therefore, when you add sodium hypochlorite, it's better to observe immediately. Once you lose the sight of the the trophy, you cannot detect it again because very, very tiny and the light microscope is not sufficient to detect. But if you follow one individual adding the sodium hypochlorite, you can definitely detect the trophy and as fast as possible take photographs or draw the camera list diagrams. Now, what are the literature? What kind of literature we have now? We have the standard literature by Coste. There are 2 volumes, and this gives you these 2 volumes are essential to identify. But this does not cover the group Deloidia. There is another book separately. It was produced by Donner, Joseph S. Donner in 1968, 65. So that is useful for identification of the Deloitte rotifers. For all monogonal rotifers, Coste is, although it is, the title says for Central Europe, but it's for the whole world. It includes also species from North America, and about 1,500 species are included. That is more than sufficient. Recently, because the coast volumes become outdated, especially for certain genera like Brachionas, Costa is not useful because he clubbed many species into some kind of variety or forms and so on, infra-specific taxa. So modern studies indicate that it is necessary to elevate them to species level. Therefore, Costa and Schill modified brought out some series of articles, but most of them are included now in the special SPP academic publishers. There are series of volumes, there are about 14, 15 volumes on not only on Rotifera, and many other groups, the Cladocera, the Copiopoda, they all have appeared, and some of them are still in process, but probably they may not see light because the chief editor-in-chief is Professor Henry A. Dumont. He has become a little less active now, and he has also some problems with his eyes. And authors are also not willing to prepare the volumes in relatively faster. Even the already published works need upgradation. That is also not done for some species, especially molecular tools have given more species. Fortunately for us, for many groups, including the Brachiones, Plicatlis alone, which is supposed to be only 2 species, the species complex of 2 species. But now we know there are at least 16 species are detected based on the molecular tools. So some of these aspects are not included in the older guides, yet these guides are useful to a great extent unless you are interested in subspecies level. These guides are sufficient. Now, for literature within India, B.K. Sharma's nearly 200 articles, mostly on taxonomy of Rotifera. A few are also on the taxonomy of Cladocera, but the majority of them are on taxonomy of Rotifera. You can consult them. Many of the articles are published in Zoological Survey of India, monographs, and some of the monographs are exhausted already. So you can depend on this. Do not directly go to Coste because you will be misled by seeing the number of subspecies, infra-specific forms, and so on. And many of them probably exist only in Europe. So B.K. Sharma's guides are useful, and there are also other authors who have produced, like Dhanapati also made some guide, identification guides. So some of these things that I'm mentioning, these can be done even in an undergraduate college. You do not need university facilities. You do not need the expensive equipment, projects, and so on. Whatever projects given to you by the UGC and CSAR, I don't know, some such organizations, the smaller budget is sufficient to develop this. One of the greatest taxonomists in the copiopoda, for example, calanoids, is unfortunately passed away, Dr. Y. Rangareddy, whom I know for the last, I know him at least 40 years, and then unfortunately passed away, but his guides are still useful. Even researchers from Europe and North America are dependent on his calanoid identification guides. So that is the power of doing the taxonomical works uh, using simple equipment. For example, camera lens is more than 150 years old model, and some of them are updated, but still the concept is the same. So these can be easily prepared in the time that you have after teaching, for example, some depending on the workload that you have. All teachers have teaching workload obligation, is independent, whether you work in an undergraduate institution or at the university. We still have to give classes, and then that takes a lot of time, but that also gives you an opportunity to interact with the students, and we explain what cannot be generally published formally in journals, articles, and so on, but you can discuss with them and transfer the information. Some of these techniques you don't find them in any single book, but they are all by experience or by reading literature part by part from different journals. You get this information. So this one part I would like to tell you. The second part I would like to show you is this. I will stop and take another one. And I share the screen for another uh, aspect. Okay, this is not needed. Let's see. Then I will go for the uh, 2 volumes of Coste. Dr. Nandini already mentioned about uh, the volumes of Coste. We have some copies of that one, but uh, the real description is like this. Okay. Now I will share the screen again.
Yes, okay. Yes, we can see the slides.
You can see it?
Yeah,
Yeah.
yeah, it's okay.
Excellent. So there are 2 volumes of Rotatoria. Old name is Rotatoria, recent name is Rotifera. It's Central Europe. It is there are 2 volumes. One volume is exclusively in the text. Another volume is simply the line diagrams. No photographs are used, so you can also consult them. The books, each volume is about $100, not really expensive. And then you can also purchase some in Amazon. Suppliers also used books there, but they are really good. Some of the retired rotiphorologists, they don't find any other person to purchase. So they sell online, and they cost about $50. And some of our students purchased them. I don't know they're still available, but the original cost of each of them about $100 each volume, and it's worth it. And we scan them because we use regularly in the class, because this is the single monograph available today, comprising of more than 1,500 species of monogonats. So that is more than adequate for any level of teaching or research, uh, unless you are more interested in subspecies level. But this book is worth to have it. Now, next part I will share. Are you able to see the figures? Okay. So these meticulously drawn these figures with only camera lucida, and These all you will see. This is the scanned version I use for the laboratory. It's in the PDF version, and on the right side you have the photos, and the left side you have the text. So the text I just copied and pasted just below the figures. All figures are drawn with simple camera lucida, not any expensive camera lucidas are available attached to the microscope. Even monocular microscopes can also be useful for this, and they are relatively cheap. The prism type, the mirror type, the tube type, there are different types depending on the cost. But even the primitive ones are sufficient to draw these diagrams. All of them are drawn with pencil, and then they use India ink, China ink, whatever it is, and then they are all drawn to the same site. So this have posted this from posted only textbook. The part of the description I have not scanned here, I have not brought it here, but this is useful for identification of all the species that you have in the world. At least vast majority you can get from this. Okay, now Dr. Nandini already also mentioned about uh, Hendrik Seger's works, so I will share that as well. This work I will give you, Dr. Nandini will send that to you. Uh, this is share the screen again. Okay, this is Zootaxa. Uh, they have issue number 1564 and annotated checklist of rotifer. This is most authentic list of species. Whenever you are writing a species, the descriptor or the author of the taxon, you will find here completely. This is the how zoogeographical distribution of the species. Um, this is used by Kuhn Martens, and then many people use for the zooplankton. This is an excellent model. All the zoogeographic regions are given here, and the species are distributed. Some are restricted to Nearctic, some of them restricted to Palearctic, some of them are Neotropical, Afrotropical, and so on. So the importance of this one is, as Dr. Nandini already mentioned, the correct species name, taxonomic stability, the author, and the synonyms of all the species of rotifers, including the daloids. So this is a very valuable contribution. I think Dr. Hendrik took to write this one at least 15 years to collect all the species and synonymize many of them. Um, this is how the species a genus, the valid species under its name are listed. And here you will also find the location where you will find them. If a species that is not found in India, for example, is found only in Europe, you should first think, consult this one. Why you find the species in India, and you can, I don't know, some kind of background noise is coming. Uh, okay. Anyway, this is how you identify all the species. You can confirm the taxonomic status of all the species. It is approved by the world taxonomists and is being used widely. For example, if you have one species, the Neuropsicoilata, and there is also Suzuki made several subspecies, and they are all synonymized here based on the observations and literature figures supported by other researchers. This is a free copy you can download easily from the internet, from the is open access. Okay, now the another aspect that I have received from Professor Das, some of the figures, how we identify them. So I will see again share the screen with you. Let me see where it is. Okay. Can you see the species? So species of this kind are difficult to identify, but by experience, I can tell you what species it is. It is the genus Lacane, species Lacane claustrocerca. What is missing here or what is not in the focus, is out of focus, is the toe. There is a toe, distinct toe here. What you are seeing here is the anterior part of the animal, that is that is a trophy and corona. Somewhere a trophy is located here. This is the corona part. I'll show you. No, this I cannot see this one, but anyway, this is the corona part has come out, so you need to add high concentration of formalin. So as I mentioned before, that 4% formalin, normally the formalin, commercial formalin is about 33%, 34%. Calculate in order to get the 4% formalin at the end, the concentration of the formalin in the sample should be 4% or even slightly lower, no problem. For lasting for more, usually you can use 5% as well. So if it is not properly, if the concentration of the formalin is not sufficient, then the animal gets expanded irregularly and you cannot identify the species. So this is one of the problems we have in when you are fixing the sample. Make sure that you add enough concentration of formalin. Then I will show you some of the figures again. Again, I have to share because I did not put all of them in the last night only. I received them, so I did not put them in the PowerPoint presentation. But anyway, I can show you some of them. Uh, okay, so again, I share the screen. Okay, I think you can see this. It's the same species. But again, another view, I still do not see the toe. The foot and toes are located. This is the foot one segment. This is the toe. Foot in Lacane is one segmented. So I don't know why it is not moving to the next level. Okay. Yeah, it is moving. So this is Deloid rotifer. It's a Philodina. The genus is Philodina. There are many species in Philodina. Philodinidae has about 108 species, so very difficult to identify them unless you have the live individual. Costae, the volume Costae does not include this group of animals. For that, you have to consult the other literature. Kutikova also made recently about 8 years ago, one volume, or you can consult also a donor's special volume of 1965, exclusively order Deloidia. In those days, they consider this as an order. That can be identifiable only with the live species and how corona is expanded. If corona is corona, how it is expanded, and foot, toes, spurs, if there is any such structure. This is the same species in another view. Also the same species. This is what we are seeing. The corona is bilobed. There are two lobes in the corona. That is useful. Here I can see one part of the corona and this is another part of the corona. It's out of focus, but there are programs, by the way, you can merge them, the different photographs into a single, like stacking they call, and that will be useful to represent all the features in a single slide. And this is another one. This is again is a species of Lacane. Possibly is also what is you can see as a very small structure is foot. The toe is located here. This again, Deloidia, the Philodina. This is Deloid. When you add formalin, they get contracted irregularly. That is why formalin is not useful for identification. Fixed species of Deloids are not identifiable because the corona is withdrawn, the foot and toes are withdrawn, the spurs are withdrawn. So it is just all the Deloids, different species of Deloids will look more or less like this. So it is not easy to identify them. Therefore, this is something you need to observe only in under the live conditions. This is another view of the same. This is This is Lepidella. You will see Lepidella has a three-segmented foot. There are three or four in some cases foot, and what you are seeing is a toes. There are two toes well separated, but in this case they are joined. But if you are using 40 times magnification, you will definitely see the two clear toes. In some cases, some individuals, uh, the toes are very clearly separated. So there is an anterior end. This is head opening is lunate, something in the form of half moon, and there is a ventral plate. And a ventral plate, you also have another opening, ventral opening. The foot opening is also semicircular. Usually, is long. So the number of segments are constant. The foot pseudo segments, they are called, they are constant for most genera. What is more important here is variable is the toe length. The toe can be length, and in this case, as I mentioned before, cross section is important. That you need to orient the animal. How thick is the animal? The dorsal plate and ventral plate, how they are. Uh, this is Lepidella patella, Lepidella ovalis look alike. The only difference is the width of the animal. Therefore, if the width is not given, it is not easy to identify. More or less, we can say is a Lepidella ovalis, but it is necessary to check the cross section. This is another species. It is the form you can see uh, more or less like a barrel. It's like gun barrel type. Is very long. Anterior part is wide, and the number of spines here, Trichotria, Trichocerca, then all these species. This another genus. Altogether, we need to see how many spines are present on the part of the posterior, and so I would
This
say
is
that.
Mytilena, no?
Yeah, I'm coming about Mytilena.
Mm.
Mytilena ventralis has got two spines, additional spines here on the ventral part. This is the dorsal part. Ventral part, there are two more are present in Mytilena. Mytilena ventralis is very common. Because the spines are absent here, I would say it is Mytilena ventralis. So, as I mentioned, Trichocerca, species of Trichocerca, they look very similar, but the difference is they also look very elongated, but they also got foot and toes, but this kind of strong spines are present only in the anterior part. So you get more or less confusion when you are checking many different varieties, genera in the same water body. So you need to have first and look at the all the figures of rotifera, so you have an idea how they look like. This is I something went wrong again. I'll check again. Um, this is Lepidella species of looks like Lepidella. Um,
1 minute.
Something has gone again. I'll put, please 1 second. Because this is gone out. 1 second. I'm trying to see this species. That was this one. No, this is we have already seen. But I'm opening with another program, that is why I'm not able to get this idea. This one? Aha, this is the one. Lepidella triptera. Lepidella triptera. So I didn't stop the screen and again share the screen. Lepidella triptera. Can you see the animal Lepidella triptera? Here you will see, when you see the animal here, you see some structure. I can see some kind of elevation. So this is Lepidella triptera. So in the costa you get this information. Many different morphological forms are there. These are not different species, but different morphological forms are there. So this also you can get in costae, but you need to draw the diagrams because in order to see how variability in the morphology can exist in this group of organisms. Any other species that I have? 1 second, let me just stop the screen. And I think these are the only species that I have from the list that I received. But if you have a species, please project on the screen. I can see them. I can try to identify them. Two species of Mytilena that you have, Mytilena ventralis, Mytilena mucronata, both are present in India. So you can get both of them. Mytilena acanthophora is also there, also common, coexist in the same tropical conditions, but it's mostly associated with the aquatic vegetation. So it is important to identify the species in order to use them for as indicators of water quality or as bioassay organism or even culturing as live species.
Excuse me, sir. Hello.
Sir,
Yes.
Am I audible?
Yeah.
Sir, we have sent you one more photo of the rotifer species that we observed, the last one.
Did you send to my email?
Yes, sir. Yes, sir. Sir, please cross check.
Yeah, I'll just check it.
Yes, thank you.
if you take nice photographs like these with your cell phone and send it to Dr. Sarma or send it to me, we can try to help you to identify them. Dr. Sarma is the final word, of course. But after that, we can also interact with other people in the world and try to help you. So please don't hesitate, especially the young people, please send us.
Yes, ma'am. Thank you so much.
Welcome.
I did not get anything here. I downloaded all the photos.
Which mail did you send it to? Did you
Sir,
get
the
it?
last one in the photographs I have
Okay.
sent.
Okay, then let me see. Yes, you're right. Yeah, okay. Yeah. I will share now the screen. Correct. So this one is, then in that case, I have to show again, share the screen and this one. Is this one?
Yes.
Yeah. This is Brachyonus angularis with a resting egg. You will see some part is darker, the other part is transparent, and it is a resting egg of Brachyonus angularis. But it is with still corona was expanded and formalin concentration was not sufficient, or it is a dead individuals in the field and you added formalin. Dead individuals who generally have this kind of trophic corona coming out of the lorica. So but I can tell you that what species it is definitely Brachyonus angularis because based on the egg structure. But I still need to see if there are any spines. There must be only two in the median spines. The size very small and two must be there. Then then it can be confirmed much better. But as of now, I can say it is Brachyonus angularis. But you still need to see the anterior spines. So you need to take photographs in different orientations. Just do not put only one orientation. Second important point is not every individual in the zooplankton collection can be identified because some of them do not have the proper orientation or they are fixed when they are dead already in the field. So morphological variations can confuse us. Therefore, you need to study more than one individual. In other words, some kind of study of a population. It also gives you the idea of the variation, the spine length, spine form, also the body size. So body size is also important. You need to put a scale to know how big is this animal. Angularis is generally very small. It is about less than 100 micrometers, about 80 to 90, whereas Brachyonus calciflorus is very big, can be up to 150 micrometers. So size determines also to some extent what species we are talking about, in addition to the spines.
Also, the presence of putting a cover slip, no, Sarma, with
Yeah.
plasticine.
Yeah. So when you compress the animal, then very difficult to identify again. So you take a cover slip and little bit of plasticine on the four sides of the cover slip and make as a platform, and then put that over that, so that the object of the microscope does not touch the specimen, but at the same time you can study the features, the morphological features of the individuals. So any other participant has any photographs that we can try to identify? If you
Hello,
have,
sir. Sir, these photographs are taken from our student, MSc students during their study. These are all living organism
ah,
in living conditions. We just put the cover slip on the water drop and then take the photographs through microscope in computer or from the mobile phone.
oh, excellent. So next time, whenever you are collecting with the students, please fix the samples in formalin, and then they will be useful for future identification.
Okay, okay. Thank you, sir.
Dr. Nandini, do you have any suggestion for them for anything?
Excuse me, sir.
She
Please.
has a question.
Okay.
Sir, please share that protocol that you shared in the slide about
Yeah.
rotifer fixation and sample collection.
Okay. Yeah.
Thank you, sir. Thank you so much.
I will send the slides to some Dr. Das. You can collect from her.
Yes, sir. Thank you so much.
I don't know if you can hear me. Can you hear me?
Yes, ma'am.
I just wanted to suggest, since we are also here tomorrow, if
Yeah.
the students are interested, they can go back to their lab, take a few pictures of fixed specimens of other rotifer species. And if they want to show Dr. Sarma tomorrow, they can also do that. But more importantly, especially for young students who are working in this field, please do take this opportunity, take lots of photographs, send it to us, and we can definitely help you to determine the species. And it would be best also if you are in contact with, I don't know, what about BK Sarma? Is he still doing anything?
I think he retired, but continuing, I think.
Okay. So maybe you can find his email address, and also you can be in contact with Dr. BK Sarma.
Okay, thank you very much, ma'am. Ma'am, can there is any particular key for the identification of zooplanktons?
There are many keys, and these keys are based on each group. So you will have keys for rotifers, keys for different families of rotifera. So you will have a lot of keys are there, but it is always very difficult to use the keys because you need to know each structure that they are talking about in the key.
Yeah.
So for that reason, please follow the guide that Dr. Sarma said. The Coste books are very useful, and any other images. And
Okay.
I think BK has done some work, no, Sarma, with
Yeah,
some
many keys are
Indian
available.
books.
Keys are
Indian,
available.
yeah.
Yeah.
So you
Ma'am,
will have, yes.
is there any site for the identification? We can send the photographs like birds and butterfly. They are doing these sites. There is any site for
There are, I'll tell
zooplanktons?
you. No, I'll tell you that there are, if you want to talk about experts who can
Yeah.
identify rotifera,
Yeah.
there are only 5 who are very reliable now, who are working.
Okay.
So there is Bob Wallace, Dr. Sarma, Hendrik Segers, Russell Scheel in Australia, and Christian Jersebek in Austria.
Okay.
So these are the only 5 people in whose identification you can actually say that, yes, it is correct. These people are very kind and they will answer your messages. But for that reason, that's why I'm telling you that there is no site as such available, but consider Dr. Sarma as a site for yourself.
Yes,
And
yes,
you can send
definitely.
him all the photographs and he will be able to help you a lot.
Okay.
I hope he doesn't disagree with me later.
No, no, ma'am. Actually, my one of my students is doing working on the zooplanktons.
Yes.
So I am asking about the identification. So I will in
Please send it
contact
to
with
us.
Dr. Sharma sir.
Yes, please be in contact with him. He will surely help you. There's no problem about it at all.
Thank you very much, ma'am.
You're most welcome.
Any questions?
Hello.
Hello.
Good afternoon, everyone. I am pleased to be stand on behalf of our organizing team. It's my privilege to propose a vote of thanks on this occasion. My heart filled with lot of gratitude and respect for our distinguished guest speaker, Dr. S.S. Sharma, their invaluable time for us to grace this occasion, but also for enlightened us with their commendable talk identification of rotifers from different sites. He also explained about the how to collect the rotifers and observe under the microscope, identification of species under external morphology and tophical morphology. For the identification of rotifers, use the different types of literature, and he also explained the zoo taxa species. Thank you, ma'am. Thank you, sir, for brief knowledge about the rotifers and excellent lectures. Thank you, sir.
We thank you. We thank you.
Thank you very much. Thank you all.
Thank you very much, ma'am.
Thank you. Thank you so much. So tomorrow we will see you now with another 2 talks, now on manuscript preparation
Okay.
and ethics of publishing.
Thank you.
Thank you very, very much.
Thank you very much.
So we can disconnect now.
Okay. Thank you,
Thank
ma'am.
you.
Rachel. We will meet
Bye-bye.
tomorrow. Bye.
Bye.
Now I request to all the participants, please gather here after the lunch.